let's jump into our Google Classroom here. Yeah, that's fine. All right, so before we begin today, just a reminder that your daily assignment for today is a little bit different. It's not an online assignment like most of your other ones will be. Instead, it's this handwritten assignment about talking about past experiences with science classes, either here at the Excel Center or elsewhere. What did you like about your past science classes? What did you not like about them? And what's some stuff that you're excited to learn about in this class? And so this is one of the final writing assignments we're going to do for the Excel Center kind of writing project that we've been working on the last couple weeks. And so because of that, it has to be handwritten on a piece of paper. It can't be typed up or texted out or anything like that. And when you turn it in, please make sure that you have your full name, so your full first and last name, and the date. So if you write it today, it would be today's date 4-8-21. So again, I'm not going to be reading it and like criticizing the spelling or the grammar or anything like that. If you turn it in to me, you get the full points, and that's all that you really have to worry about it. So again, please do write this up and get it turned in if you get a chance, either today or if not today, sometime later on this week or this weekend. I really, really do appreciate it from everybody here at the Excel Center. So anyways, for the actual notes that we're going to be talking about today, we're going to be going over something called the scientific method. You may have heard of this before, you may not have. Essentially, it is sort of a basic series of steps that scientists use in order to observe and study the world around them. So scientists don't just throw out random questions into the universe and then come up with experiments in order to test those. There is a process by which science happens, by which new discoveries are made. And we call that process the scientific method. So there are a series of steps in the scientific method, and we're going to go through them kind of one by one today. But just to kind of give you a little bit of an overview of what they all are, we have you make observations. You use those observations to come up with a question. You use that question to develop a hypothesis, a type of educated scientific guess. You then do an experiment in order to test your hypothesis. You gather some data from your experiment. You take some measurements. You write it all down. You use the data that you collect, your results, to come to some sort of conclusion, some sort of final idea about what is going on with your system that you're studying. So all scientists use the same basic method, same basic kind of line of questioning in order to develop their experiments and come up with answers to the questions that they have about their world. So it all starts off with observation. Scientists observing something either in another experiment or something in their own life or their own house or their own outdoors. And observations are gathered through the senses. Specifically, all 5 of your senses can count as observations. So normally when we think of observations, we think of seeing things, right? You think, oh, if you observe something, that means that you saw it with your eyes. And yeah, that is one way to take observations. That's probably the most common way, honestly, since as humans, we are very visual species. We use our eyes to process a lot of stuff. But that is not the only way that you can observe something. You can tell a piece of fruit has gone bad if you smell it and it smells really gross, right? So you can smell in order to observe the world around you. You can touch, so I can feel that this table and my computer are super smooth and made out of hard plastic. You can hear, so you can tell if a noise is louder or quieter, or you can tell the direction that a noise is coming from. Those are observations that you can make with your ears. And you can also taste, right? When you taste something, you can tell, is it sweet? Is it sour? Is it bitter? Does it taste fresh? Does it taste rotten? Does it taste gross? Any of your senses can be used to gather observations about the world around you. But again, in science, we're noticing something about the natural world. So science is all about trying to come up with answers to things that are happening out in the natural world. So for example, one example that I'm going to kind of be going back to as we go through these to kind of explain all the different steps. Imagine that you are out walking through like the forest, right? You're on some sort of nature trail and you come across this pond that has a bunch of salamanders in it. If you don't know what a salamander is, it's basically like a type of lizard that likes to live in and around water. So they usually have to be close to some sort of water source, and that makes them salamanders. So most salamanders, like most lizards, their tail is one like kind of big, long, straight line coming off the back of their body. It might be long and thin, it might be kind of big and chunky, but for the most part, it's a big straight tail. However, while you are on your nature walk, you notice that all of the salamanders around this particular pond all have weird curved tails. Their tails have a bunch of like twists and turns in them. And you think, that's not normal. Salamanders are supposed to have long, straight tails. The ones in this particular pond have these weird curved tails. That is unusual. That's not something that you see every day. So that might lead you to a hypothesis or a guess as to why these salamanders have curved tails, right? You see the weird lizards, the weird salamanders. You say, why are they like that? And in order to try and answer that question, you come up with a hypothesis. So a hypothesis is a kind of like a prediction. It is a suggested solution to the problem. It's sort of like a scientific guess, if you will. It must be testable, so it must be something that you can do an experiment on in order to come up with a good hypothesis. And so that means that you can't blame like the gods for having altered these salamanders this way, or you can't blame like the alignment of the planets when they were born 6 years ago on the second Sunday in June. That's not stuff that you can really test as a scientist. So a hypothesis, your guess, your prediction has to be something that you can test. Sometimes hypotheses are written as if-then statements, meaning if I do an experiment, then these are the results I think I'm going to get. Right? So if I add fertilizer to my plants, then I think the plants will grow tall, taller than other plants. So you're making some sort of prediction when you develop your hypothesis. You're guessing what you think, if you do an experiment, what you think the result of that experiment would be. So, for example, you might hypothesize on your nature walk that the salamanders in this pond have weird curvy tails because there's some sort of pollution in their pond. So if salamanders are exposed to pollution, then they will end up having weird curvy tails. So that's an example of a hypothesis. You made an observation you saw the weird salamanders, and then you came up with some sort of prediction, some sort of guess as to why their tails were growing so funny, why they looked different than how a normal salamander should look. So you have your observation, you have your hypothesis, your guess. Now it's time for you to test this hypothesis. So in order to do that, you're going to need to do some sort of experiment. So an experiment is something that you can do to test your hypothesis and see, was your hypothesis right or was your hypothesis wrong? Did you make the right guess, or was it something else that's causing this problem? So when you are setting up an experiment, there are some different things called variables that you need to be aware of and ready to control. So variables are factors in the experiment that can change. So if you're setting up an experiment to test if pollution is causing the salamanders to have weird tails, then the variables in the experiment are all the different things that could be done to change how the experiment works. And there's kind of three major types of variables that we're going to talk about here in a second. So in order for an experiment to be a good experiment, also known as a valid experiment, there has to be only one variable that you change, only one thing that you as a scientist purposely change as part of the experiment. So the reason for this is, I'll give you an example and see if you can pick out what a good reason is. So say you're doing your salamander experiment. You want to see if pollution causes salamanders to grow up with weird curvy tails. So you set up two different experimental kind of areas, two different experimental rooms. You put a whole bunch of baby salamanders in each room. And then in one room, you keep it at 78 degrees. You keep it at like 80% humidity because salamanders really like it when it's humid. You provide a big pond in the middle of the room for the salamanders to access. And you make sure that all of the soil and stuff that they're living in, all the rocks that they're dealing with, do not have any pollution on them. So one nice warm area, no pollution, lots of water, lots of humidity. You set up a second chamber where you keep it at 60 degrees Fahrenheit, so a little bit cooler, a little on the kind of chilly side. You restrict the amount of water in there, so it's a lot less humid and there's not as much water available to the salamanders on the ground. And you add in the pollution to that second chamber. So you do your experiment, you have all these variables set up, and then at the end of the experiment, the salamanders in the first warm water, no pollution chamber all have straight tails, and the salamanders in the second chamber, the colder, less water, pollution chamber, they all have curvy tails. So based on your experiment, did you prove that it was the pollution that was causing the tails to grow all crooked? So feel free to either answer out loud or put it in the chat. But based on this experiment where you have different temperatures, different water levels, and different amounts of pollution, is that a good way to test and see if the pollution causes the tails to grow weird?
Yes.
All right, we got a couple yeses. We got Alexia saying you can't know because everything was different. Alexia, you have hit it right on the head. So the answer is that it is not a good experiment because you don't know that it was the pollution that caused the tails to grow weird. Maybe salamanders get curvy tails when they grow up in a cold environment. Maybe salamanders get curvy tails when they're restricted with their water access. Because you changed so much stuff in your experiment, you don't know what it was that you changed that caused the salamanders to grow with the weird tails. Just like Alexia was saying, everything about that second chamber was different. So you don't know which variable it was that caused the tails to change. However, on the flip side of that, if you had two chambers that were the exact same, they had the same amount of salamanders, they were kept at 70 degrees, they were kept at 80% humidity, they each had plenty of water. But in one chamber, it was pollution-free. In one chamber, you added in pollution to the soil. In that experiment, if the pollution chamber had the salamanders with the weird tails, then you would know that it was the pollution that caused that problem. Since the pollution was the only thing that you changed, and the salamanders grew up different in the pollution chamber, that would be really good evidence that the pollution was the cause of the curvy tails. Yeah, Anthony, question, go for it.
So basically, the more things that you change, then like the more like those different things can cause like you don't like if there's only one thing that changes, you know that one thing did it. But if there's multiple things that changes, you have to like try to figure out which one of those specific things changed it. So it makes it like way more complicated.
Mm-hmm. Yep, exactly. That is 100% the point of this slide right here, is that if you only change one thing and you see a result, you know that that result was due to that one thing that you changed. That was the only thing that was different, so that must be causing the change. However, if you change a whole bunch of stuff all at the same time and you end up with the result, then it's like, well, it could have been the pollution, but also maybe it was the temperature, or maybe it was something about the water that they had. Maybe they needed more water than what they had. You don't know for sure. You don't have enough information to answer the question. Like Anthony said, it makes it way more complicated to figure out. So if you want to have the clearest results possible in your experiment, there should only be one thing that you are changing. Exactly. So because it's science, we always have certain words for the different types of variables that are in our experiments. So there are three major different types of variables. There are control variables, also sometimes just called controls. And then there are independent and dependent variables, which we'll get into in a second. So we're changing one thing. Again, if we want to have a good experiment, we can only change one thing. And then as the experiment goes on, we're observing or measuring what happens. So in our salamander experiment, we're observing our salamanders to see if they develop the crooked tails. If we were doing an experiment where we wanted to see what was the best plant fertilizer out there, we'd be measuring the growth of the plants to see which one grew taller or produced more flowers or stuff like that. That would be our kind of like our results, the things that we're observing or measuring as the experiment goes on. So the thing that is changing is called the independent variable. So the independent variable is the thing that we as the scientists are changing in our experiment. Like Alexi and Anthony were just telling us, you can only change one thing if you want your experiment to give you the most clear results. So that one thing that you change is called the independent variable. So either the amount of pollution that you give to the salamanders or the amount of fertilizer that you put into the plants, that would be your independent variable. That is the thing that we, as the experimenter, we as the scientists are changing. So it's called the independent variable because it does not depend on any of the other variables in the experiment. It only depends on us. So it's independent of all those other kind of natural variables. So we are changing the independent variable. The thing that we are measuring or observing at the end of our experiment, basically like our results of the experiment, are the dependent variables. So if we did our salamander pollution experiment, the dependent variable would be the number of salamanders that grow up and have crooked tails. If we did the fertilizer in the plant experiment, then the dependent variable could be the height of the plant. It could be how many leaves are produced or how many flowers are produced on the plant. But it is the thing that you are measuring, the thing that you are collecting for the results of your experiment. So it's called the dependent variable because it depends on what we do with the independent variable. So however we change the independent variable, the dependent variable should change because of that. If our hypothesis is right, if our guess as to what's going on is right, then changing the independent variable should change our dependent variable. So independent, so it's like the independent is like what we are changing at the start of the experiment. And then the dependent is what we are measuring either as the experiment is going on or like at the end of the experiment. So we have our independent variable, which is the thing that we are changing. We have the dependent variable, which is the thing that we are measuring. Then we have a lot of other variables that make up the experiment called control variables. So control variables are the things that we keep constant, we keep the same across all of our experiments so that they will not affect the outcome. So in our salamander experiment, if we wanted to test pollution and how that changed the tails, our control variables would be things like the temperature of the chamber and the humidity that was provided and the amount of water that was given to the salamanders. If we were doing an experiment with fertilizer and plants, control variables would be making sure that we use the same type of plant in all of our trials and that they're getting the same amount of sunlight and the same amount of water each day, that they're put in the exact same type of soil and the exact same size pot, because all that stuff could affect your final results, but you don't want it to. You only want the independent variable to be affecting those results. So your control variables are all the stuff that you try to keep the same across all of your experiments, all of the different trials that you're doing. So you're changing how much fertilizer each plant gets, you're measuring how tall the plant goes, and you're making sure that all your plants get the same amount of sunlight and water and are in the same type of soil. If you're doing the experiment with the salamanders. You're changing if they're exposed to pollution or not. You're measuring how many grow up with straight tails versus crooked tails. And you're keeping the temperature, the humidity, and the water level and the size of the enclosure all the same across all of your experiments. So control variables are things that you keep the same, that don't change over the course of your experiment. So again, our control variables are not being tested. We don't want to know how sunlight affects plant growth or how humidity affects the salamander tails. We're not interested in that. We don't care about that. We want to know about fertilizer or about pollution. So we keep these controls the same so that it's really easy to compare our non-pollution versus our pollution group, or to compare our non-fertilizer versus fertilizer group. If all of the controls are the same, then it makes it really easy to compare them and see, did the fertilizer have an effect on the plant growth? Did the pollution have an effect on the salamander tails? So these controls, we keep them all the same so that, just like Anthony was saying earlier, it makes it a lot easier and simpler to figure out what is causing the change, what is causing that dependent variable to be different. All right, so I'm going to have you guys help me through a kind of basic example here, where I'm going to lay out a really simple experiment type scenario for you. And then I want you guys to tell me, what is our independent variable, what is our dependent variable, and what are some of our control variables. So we're going to do an experiment. And in our experiment, we want to figure out what is the fastest way to walk home from school. So we're going to have to pretend like we're going to school like we did in the normal times when people would come on campus, go to their class, and then afterwards go home. So this example is walk home because it's, I guess, for people whose school is close by. You can also think of it as driving home, works the exact same way. So you want to know, what is the fastest route to drive from the school to my house? So normally you have like the one way that you go, right? Kind of like the main road that you take. You think maybe taking some side roads or maybe taking a different set of highways will make you get home faster. So you're going to try taking several different routes home. Maybe one day you turn right and then go straight and then turn left. Maybe one day you turn left and then go straight and then go straight and then go right. So you're going to change up how you get home, like each day a week. So Monday through Thursday, you come to school, and then afterwards, you take a different way home. And when you're doing this, you're going to time how long it takes you to get home. So as soon as you leave the building, you're going to start your stopwatch. And as soon as you pull in to your apartment building or to your driveway, you're going to stop. And so you're going to compare how long did it take you to get home on Monday versus Tuesday versus Wednesday versus Thursday when you went a different way each day. So because this is specific to you and you going to your house, you're going to make sure that it's you that is doing the driving each day. So you're not going to have like your friends come in your car and try to help you, because they might drive a different way than you do. They might be like faster or slower. And you're going to take the same car each day. So it's not like on Monday and Tuesday, you're going to drive your normal car. But then on Wednesday, you're going to borrow your brother's car. And then on Thursday, you're going to borrow your uncle's motorcycle. You're going to use the same car each one of the days. So it has the same like speed, the same acceleration and deceleration, all of that good stuff. So what are our variables in our getting home from school experiment? What is our independent variable, our dependent variable, and what are some control variables? Feel free to answer either out loud or in the chat.
I think independent traffic, maybe. Oh no, because you can't change traffic. Never mind.
Okay, so we got Alexia saying independent is the route that you take to get home. Anybody agree, disagree with that?
I agree. That sounds right.
I agree.
Cool, cool. What about the dependent variable or some of the control variables?
Dependent is like the time it takes with that specific route, right?
Mm-hmm. So yep, dependent is what you're measuring. So it's how long that it takes to get home. And then what are some of the control variables? What are some of the things that you're keeping the same between all of your experiments?
Like the time you leave and like how fast.
Mm-hmm. Yep, the time that you start. So yeah, you're leaving school at the same time each day. That's a good control variable. Let's see. Alexia says, who is driving? So yep, so it's only, you're the only person that is driving in the experiment. So it's the same person driving each time. So that's a good control variable.
What about something you can't control, like traffic?
So traffic, that would be one of those things where you wouldn't, it would be essentially out of your control. So that'd be something that could add some variability into your experiment. So hopefully by leaving at the same time each day, that helps to cut down on that a little bit. Something else that you can do in order to help control that variability is to do the same route multiple times and take kind of like an average time, right? So maybe instead of doing Monday, Tuesday, Wednesday, Thursday, and only having 4 different data points, you say, okay, week 1, I'm going to drive route A home, and I'm going to do it 4 times. And then week 2, I'm going to take route B home and do that 4 times. And then week 3, I'll do route C home and do that 4 times. That way you can take kind of an average, and that might help to reduce some of the variability that you get from, stuff you can't control, like traffic or accidents or stuff that happens out on the road. All right, so awesome. Great job. So yeah, changing up the route, how you're getting home, is the independent variable. That is the thing that you, as the experimenter, are changing in the experiment. You're changing how you get home. The time that it takes is the dependent variable. So that is what you are measuring in your experiment, right? Each day you're measuring how long it takes you from starting at the Excel Center to get to your house. And then finally, keeping the same walker, or in our case, the same driver and the same car, and leaving at the same time. Those are all control variables. Those are all things that you're going to do your best to keep the same across all of your different trials, all of your different experiments. All right, excellent job, everybody. Does anybody have any questions so far about the independent variable, dependent variable, control variables, or the stuff about the hypothesis and coming up with your prediction or your question? Any questions about anything that we've gone over so far? All righty. If not, we'll keep moving right along here. So kind of going hand in hand with the dependent variable is your data. So your data is your results of the experiment. It is what you are observing and measuring and calculating as your experiment is going on or at the end of your experiment. So there are kind of two major categories of data. You can have quantitative data, which is things like numbers. So measurements, amounts, numbers, the number of salamanders that grew up with weird tails, the height in centimeters that your plants grew, the time in minutes and seconds that it took to get home. All of that is quantitative data. You can also have qualitative data, which is observations that don't deal directly with numbers. So like if you see things change color as your experiment goes on, that'd be qualitative data. Or if you're doing some sort of like tasting experiment and you're having people rate food that they're eating, that would also be qualitative data, right? You can't really measure how sweet or how salty or how bitter that something is. So data in the form of like a description or an observation is qualitative. But for the most part, most types of data that scientists like to gather is quantitative data dealing with numbers. So you take all this data, you take all your results from your dependent variable, you put it together, and you come up with your conclusion. So your conclusion is the answer to your hypothesis based on the data that you got from your experiment. And so, did the pollution give the salamanders curved tails? If it did, then you can conclude that your hypothesis was correct and that the pollution is causing salamanders to grow with weird tails. If you added a whole bunch of fertilizer to your plants and they grew just the same amount as the plants that just got water and sunlight, then you can conclude that your hypothesis was not supported, and it turns out that the types of fertilizer that you used did not help the plants grow that you used it on. So a good conclusion will state the final results of the experiment and whether or not the hypothesis was supported. So it's essentially saying, here is what I found when I did my experiment, and it supports my hypothesis because the pollution chamber had more salamanders with crooked tails. Or my hypothesis was not supported. I took a whole bunch of different ways to drive home, and it turns out that all of them take about the same amount of time. There's not any one route to get home from the Excel Center faster than the one that I normally do. So the conclusion is all about either supporting or not supporting your hypothesis. Basically, as a scientist, you're either saying, hey, my guess was right, I followed my instincts and I was correct about that observation I made earlier, or whoops, I thought my guess was right, but the results say that my guess was not supported. I did not guess correctly. I'm going to have to go back, redo my experiment, and maybe get some data, or maybe come up with a new hypothesis, a new prediction based on the data that I got from this experiment. So if you want to make a really, really strong case for your data and for your experiment, just doing it one time and being done isn't going to give you very strong results. Because in science, especially in like academic science, people will always come at you like, oh, well, maybe it could have been this thing, it could have been this thing, or what about this thing that you didn't think of? Any one of these things could have interfered with your experiment when you did it on that particular day. So in order to make sure that the results you got were good, valid results, the best thing to do is to do experiments multiple times. Kind of like Anthony was saying earlier, you can't always depend on traffic if you're driving home from school. Usually it's about the same, but some days it's a lot better and some days it's a lot worse. So just doing the driving home from school test on a particular route on one day, maybe that day you hit a particularly bad batch of traffic and it took you longer than it might normally have. In order to get really good results, the best thing that you can do is do multiple retests, multiple copies of the same experiment. In order to show, hey, I did this thing 20 times, and on average, it took me 5.5 minutes to get home from school. Sure, 1 or 2 days was a little bit longer than that, but on average, this is how long that it took. And so by doing the experiment multiple times and getting the same result every time, it strengthens your experiment and makes your hypothesis seem more and more correct. Anthony, do you have a quick question in there?
Oh yeah, so basically like retesting is just to like you said, like it's like you're doing like an on average thing of like a time taken. It basically just like makes it more accurate and more like credible, like
Mm-hmm.
the results.
Yeah,
Oh, okay.
exactly.
That's good.
So like if you say, hey, I did this thing one time and this is the result I got, people will be like, oh, okay, that's pretty good, I guess. But if you're like, hey, I did this thing 100 times and on average, this is the result that it got. It's like, oh, wow, okay, that really must be really close to the actual final result. And so just because something happens once may not necessarily mean that it happens again. So the more similar results that you can get from multiple experiments, the stronger that your data will be and the stronger that your conclusion will be. All right, great question. So if we go back and think about all the different steps in our scientific method and kind of the way that scientists approach thinking about how to come up with experiments for the world, we identify a problem by making some sort of observation, right? You see that salamanders in the wild sometimes have crooked tails. So you might observe the type of salamander that it is, the type of area that they're living in. And those observations will lead you to a hypothesis. I believe that pollution is causing the salamanders to grow up with the crooked tails. And if they are exposed to a certain level of pollution, then they will develop these weird curvy tails. So your hypothesis, your best guess, best prediction about what is causing the problem. You can then do your experiment. Set it up to make sure you only have the one independent variable that is changing, that you control all the other variables that you can in your experiment, and then figure out what you're going to measure as your dependent variable. In this case, you're going to measure the number of salamanders that grow up and have crooked tails. You collect your data, so you follow your salamanders over the course as they grow from baby salamanders to adults, and you record how many of them and on each day you find with the crooked tails. And use that data to form a conclusion. You say, in the non-pollution chamber, I only had 1 out of 30 salamanders that developed a crooked tail, but in the pollution chamber, I had 24 of the 30 salamanders grow up with a crooked tail. That to me suggests that my hypothesis is correct and that pollution is most likely causing these tail problems in these salamanders. And then finally, if you want to really, really make sure that your results are correct, you can retest, do it again, do it multiple more times, or increase maybe the number of subjects that you tested it on. And that way you can make sure that you have the most reliable data possible. So a really good example of this retesting is with the COVID vaccine that is on the rollout right now, right? Everybody about a year ago was scrambling to begin looking for COVID vaccines. And in order to get a vaccine on the market, drug companies have to go through what's called phase 3 clinical trials, which is where they test the vaccine on thousands and thousands of people to look for two things. Does the vaccine help prevent you getting infected from COVID? And are there any major bad side effects from getting the vaccine injected into you, right? If they did it on 2 people and said, look, these 2 people both didn't get COVID and they both had no side effects, you'd probably still be like, yeah, but 2 people seems like kind of a small sample size for there. A phase 3 clinical trial requires drug companies to test on thousands and thousands of people, like tens of thousands of people are recruited to go through these drug tests. That way they have a really, really, really high chance of proving the effectiveness of their vaccine. And just in case there's like a 1 in 1,000 chance that somebody gets a really bad side effect, the more people that you test, the more likely you'll be able to find the sort of rare side effects that may not happen in everybody, only a really small number of people. So that's where this retesting and getting really, really big, what are called sample sizes, or the number of experiments that you do, comes in really handy. Vaccines have to go through that thousands and thousands of people phase 3 trial and prove that they're effective and safe before the government will let them be rolled out and start to be handed out to the general public. So that is the scientific method in a nutshell. The basic kind of series of steps that scientists try to follow when developing a solution to some sort of problem in the natural world that they are interested in. All right. Does anybody have any final questions about any of these different steps of the scientific method, from the hypothesis to the 3 different variables and experiment to collecting data and forming a conclusion? Any questions relating to the scientific method that I can help you out with here today? All right, doesn't sound like it. Sounds like everybody's pretty okay with the scientific method so far. So that is awesome. Again, that will just about wrap it up for the notes that we have today. So you have a little bit of extra time left over at the end here. Again, please remember to get the writing sample assignment written up and turned in to me today. You can either upload it to the Google Classroom, send me it, text a picture of it to me, email a picture of it to me. All of those work. I know a couple of people have done it already, which is awesome. If you haven't done it yet, please do so. I would really, really appreciate it.